Today in class, we learned that the information for our traits is contained in our DNA. The DNA is a sort of cookbook, and every recipe is a gene. Genes contains the information to produce protein. The language of DNA is made of 4 letters or bases, A, C, G, and T. This is the universal language of life and is spoken by everything creative on the planet.
Experimenting in class extracting DNA. First we have a cell, and the membrane of the cell is hydrophilic and hydrophilic. The hydrophilic loves water and the hydrophobic hates water. So we did an experiment with bananas, strawberries, and peas. With this, we add detergent to eliminate the membrane. Then with the DNA plus water plus protein plus lipid have now more a membrane. After that, the DNA precipitates with the salt, and after the DNA with alcohol clump together, and with that we can see the DNA. How a cell creates proteins. DNA is like a book with a lot of information. The genes are the sentence of this book, which contains the information to produce a protein. Proteins are the building block of life and can have different functions. For example, give us curly or straight hair. In the cell nucleus, the DNA is copied into RNA in the process named transcription. The RNA goes to the cytoplasm, where it goes into the ribosome that translates the RNA into amino acids. Amino acids are the building block of proteins. Electrophoresis, working at the laboratory. Electrophoresis is a process that is usually used in laboratories to separate DNA fragments of different lengths. This one moves from the negative to the positive pole because DNA have a negative charge. The electrophoresis have 3 steps. The first one is cast the gel. The second one, load the DNA sample. And the third one, and finally, is the run. DNA moves from the negative to the positive pole. Epigenetics. Epigenetics come from epi, which means something on the top of the DNA, and genetics, that mean genes. The epigenome has the property to turn genes off or on. To turn genes on, you have to open the histones to make the DNA readable. To turn genes off, you have to close the histones to make the DNA unreadable. The gene expression can be turned on or off depending on the environment, for example, your diet. Usually the epigenetic tags put before our birth are permanent. The epigenetic tags put after our birth are usually reversibles. This is, for example, if you drink a lot of beer, you will turn on the genes to break down alcohol. But if you stop drinking, these genes will be off and you will not be able to tolerate